Guys, it is quite late in the night, but I've decided that since the Form 4s are about to start their examination, let me complete my series in biology of the data questions. I've only left one now, 2015. I'm now going to do 2014. 2016 is repetition of 2009. So let's go to this data question and we see what they wanted. So this data question is bringing the idea of the relationship between a plant and symbiotic bacteria in root nodules. As you can see, this introductory diagram. So you can see there, we have a bean seedling, then we have the nodules, root nodules. Photosynthesis is taking place of photosynthetic products, and then we have nitrogen compounds. And also, we have nitrogen from the air there. Okay, so let's go on and see what goes on. The plant gives the products of photosynthesis to the bacteria. The bacteria uses their energy to incorporate nitrogen from air into compounds which are usable by the plant. So there is another diagram there, symbiosis. So the relationship between the root nodules and the bacteria is known as symbiosis, and the beans are known as legumes. So symbiotic relationship between legumes and Rhizobia. And you can see this diagram there. You can see the area eaten bacteria cell in the root nodule, and you see what the bacteria does. There is an enzyme known as nitrogenase, which helps convert, which helps to fix nitrogen to ammonia. All right. This nitrogenase is, I think, produced by the Rhizobia in the root nodules. So ammonia is converted to amino acids and urides. Okay. Then the amino acids are able to be converted to nucleotides. All right. And you can see the process, the legume there, the carbon dioxide, photosynthesis, and then organic acids. So let's proceed and see more. So, Rhizobia and legumes. Nitrogen is the most commonly limiting nutrient in plants. Legumes use nitrogen-fixing bacteria, specifically symbiotic Rhizobia bacteria within their root nodules to counter the limitation. Rhizobia bacteria convert nitrogen gas to ammonia in a process called nitrogen fixation. Ammonia is then assimilated into nucleotides, amino acids, vitamins, and flavones, which are essential to the growth of the plant. The plant root cells convert sugar into organic acids, which then supply to the Rhizobia in exchange. Hence, a symbiotic relationship between Rhizobia and the legumes. Have you ever wondered why some plants grow faster in certain conditions? The secret lies in the air they breathe and the nutrients they absorb. Stay tuned to discover how tweaking the carbon dioxide around plant supercharges their growth by boosting nitrogen uptake. In this video, you will not only grasp how carbon dioxide affects plant growth, but also learn practical ways this knowledge can transform farming, increase crop yields, and help tackle global food security. Master this concept and see biology from a whole new perspective. As we explore nitrogen uptake and carbon dioxide levels in plants, we will connect this scientific knowledge to real-life agricultural practices. Whether you are a student, teacher, or simply curious about the natural world, this video will equip you with insights that go beyond the classroom and into solving some of today's pressing global challenges. An experiment was done to determine the uptake of nitrogen for the soil by broad bean seedlings. The experiment was done with one set of seedlings M, grown in the atmosphere, enriched with carbon dioxide, and another set of seedlings N, grown in normal atmosphere. The amount of nitrogen in each seedling was measured in milligrams at intervals of 10 days. The table below shows the results obtained. So that is the table. That is the table. The table is there. Okay. I'm trying to fix this. Okay. So this is 2014 Biology Paper 2 data question. And the question was number 6 and the marks was to 20. So from the table you can see set M and set N, amount of nitrogen in milligrams and time in days. So the experiment was done for 105 days. Using the same axis, draw line graphs of nitrogen uptake by 2 M and N sets of broad bean seedlings against time. There you are given 8 marks. Determine the rate of uptake of nitrogen in set M between 65 and 85 days. What is the relationship between carbon dioxide concentration in the air and nitrogen uptake? Account for the relationship in C1 above. What would happen to the concentration of nitrogen in the seedling in set M if after 75 days the seedlings are transferred to a normal atmosphere? Explain your answer in D above. State 3 ways in which nitrogen fixation occurs. Let's now come to drawing the line graphs of the nitrogen uptake by 2 sets of broad seedlings, M and N, against time, and we follow the following steps. The first step is to create a table where you rearrange the data for easier reference when plotting. There you are, rearranging the data for each reference when plotting the graph. You can see time, days, set M in milligrams, set N in milligrams. Step number 2, plotting the graph. X-axis is horizontal, represent time in days from 15 to 105 days. Y-axis vertical represents the amount of nitrogen in milligrams. Choose a scale that accommodates values up to 915 milligrams. Plot points for both set M and set N on the same axis. For set M, mark the points using one symbol, e.g. dots. For set N, use a different symbol, e.g. x-axis. But I said, as a mathematician, you are not going to use those dots. Number 3, connect the points. Draw smooth curves or straight lines, connecting the points for set M and set N. Ensure you label both lines to distinguish between set M and set N. Once plotted, this graph will show how nitrogen uptake differs between seedlings grown in carbon for oxide-enriched atmosphere and those in normal conditions. To draw the line graph for the nitrogen uptake by the two sets against time, follow these steps. Materials needed is graph paper, a pencil, a ruler, colored pens or pencils or pens to differentiate the two sets. Draw the axis, horizontal axis. All right. Choose a suitable scale such that you can fit the ridge comfortably on the graph paper. For example, 1 centimeter could represent 10 days. Vertical axis, label it amount of nitrogen and scale it based on the nitrogen uptake. Values 0 to 915 milligrams for set M and 0 to 140 milligrams for set N. You could use a scale such as 1 centimeter equals 100. Use the data points provided for set M. For example, on day 15, x equals 15, the amount of nitrogen is 0 milligrams, y is 0. Mark this point on the graph. Continue plotting points for set M. And you can see all the set points there. After all points are plotted, connect them with a smooth line. Now plot the data for set N on the same graph. For example, on day 15, x equals 15, the amount of nitrogen is 0, y is equals 0. Mark this point on the graph. Continue plotting the points for set N, and there you have the points. After all points are plotted, connect them with a smooth line. Add a title to the top of the graph, nitrogen uptake by broad bean seedlings, set M and N over time. Label the two lines of the graph. You can use a different color or label the lines directly as set M and set N. Ensure the axes are labeled as mentioned in step 1. If using different colors for the two sets, create a small box indicating the color used for each set M and set N. So as is the graph, all right, with the heading there, line graphs of nitrogen uptake by two sets of broad bean seedlings against M. Of course, x-axis time in days, y-axis amount of nitrogen in milligrams uptake by M, and N seeds. Then you identify the graph set M and set N. Marking of the graph. The curve must be continuous. Graph must be 3/4 of graph paper. Axis reversed or inverted graph. Maximum is 1 mark for identification only. 2 separate graphs, mark first one, maximum 5. And the marks are distributed as scale 1, plotting 1, identification 1, axis 1. No, axis 2 marks. Plotting P 2 marks. Scale 2 marks. Curve 1 mark. Identification 1 mark. Penalize 1 graph with a double line. Only 1 graph is give for A, give 4.5, you round off to 5. Extrapolate. Maximum 2 small squares for the curve. Here is the graph showing the nitrogen uptake by 2 sets of brood seedlings over time. So this is generated. The blue line represents M, while the red represents N. Okay. The graph demonstrates that set M experienced a significantly higher uptake of nitrogen compared to set N over the course of the experiment. So that is the computer-generated graph. You can see it's just shaped like the other one, but you can see the key there. The blue one is set M, which is enriched with carbon dioxide, and set N normal atmosphere. To determine the rate of uptake of nitrogen in set M between 65 and 85 days, 85, you have 80-60, 65, you have 395, 80-60 minus 395 is 465. The rate is changed over time, which is 465 over 20, which is 23.25 per day. So this is the way this is generated from the computer. To find the rate of nitrogen uptake, we use the formula rate of uptake. Okay, change in nitrogen, change in time. And from the data you can see set M 395, set N 8060, set M, yes, in both cases. So the rate of uptake is 65 and 85. This is 60 over 85 minus 465 over 65, 465 over 20, 23.25 milligrams per day. The rate of nitrogen uptake in ZM between 65 and 85 days is 23.25 milligrams per day. So you can compare the examination council answer and the one I generated or researched. Relationship between carbon dioxide concentration in the air and nitrogen uptake. The higher the carbon dioxide in the air, the higher the uptake of nitrogen by the plant. Accounting for the relationship in C above, carbon dioxide is utilized by the plant to carry out photosynthesis. Manufactured foods, e.g. glucose, are carried to the root nodules for use by Rhizobia bacteria. Increased number of Rhizobia bacteria increases the rate of nitrogen fixation, where atmospheric nitrogen is converted into nitrate for use by the plant. What is the relationship between carbon? Okay, this is the data. So what I researched again. So the first answer is for examination council. The data shows that seedlings in set M grown in an atmosphere enriched with carbon dioxide absorbed more nitrogen compared to seedlings in set M grown in normal atmosphere. This suggests a positive relationship between carbon dioxide concentration and the nitrogen uptake. As the carbon dioxide concentration increases, nitrogen uptake also increases. Account for the relationship. The increased concentration of carbon dioxide enhances the rate of photosynthesis in plants. Plants can grow faster and develop more tissues, which in turn require more nitrogen. Nitrogen is essential for the synthesis of proteins and other compounds for critical growth. So its uptake increases as a result of the enhanced growth stimulated by higher carbon dioxide levels. So examination council. What would happen to the concentration of nitrogen in set M after 75 days? It would reduce. Explanation. Reduced carbon dioxide concentration results in lower rates of photosynthesis, resulting in reduced food. Less food materials available to symbiotic bacteria lowers their activities. This lowers the rate of fixing or converting nitrogen to nitrate, a usable form by the plant. For the digital research, if set M seedlings were transferred to normal atmosphere after 75 days, the rate of nitrogen uptake would likely decrease. Explanation. The decreased rate of nitrogen uptake would occur because transferring the seedlings to normal atmosphere would reduce the concentration of carbon dioxide available for photosynthesis. With less carbon dioxide, photosynthesis would slow down, resulting in reduced growth and hence less nitrogen being needed for photosynthesis, protein synthesis, and other growth-related functions. So it is up to you to choose your best answer. E. 3 ways in which nitrogen fixation occurs. The examination council answer. Nitrogen fixation by symbiotic bacteria, nitrogen fixation by lightning, and nitrogen fixation by free-living microorganisms. Research. 3 ways. One is biological nitrogen fixation. The other one is industrial fixation or Humber process. And the third one is atmospheric fixation. So you can see here I've organized it a laboratory. And in biological nitrogen fixation, it is carried by nitrogen-fixing bacteria such as Rhizobia in legume root nodules and free-living bacteria such as Asobacter. These microorganisms convert atmospheric nitrogen into ammonia, which can be used by plants. For industrial Humber process, is an artificial process where nitrogen gas from the air is combined with hydrogen and high pressure and temperature to form ammonia, which is then used to produce fertilizers. So that is the way we produce fertilizers. Atmospheric nitrogen is fixed by lightning, which provides the energy to split the nitrogen molecules in the air, allowing them to combine with oxygen to form nitrogen oxides. These compounds dissolve in rainwater to form nitrates, which are absorbed by plants. Real-life application. Agriculture practices. Use of carbon enrichment in greenhouses. Fertilizer application timing. Impact of rising carbon 4 oxide levels. Research on crop resilience. Sustainable agriculture, legume crop rotation. Environmental conservation. Reducing nitrogen runoff. So I end by saying, by applying knowledge from this experiment, agriculture practices can be optimized for both productivity and sustainability, helping ensure food security while reducing environmental impact. Acknowledgement, special thanks to my teammates during marking exercise. Creative Commons Attribution, Share Alike 4.0 International, Authors, Neflanas, 13th July 2019, root non-use, and Joyrine Chepkori, 17th December 2016, Symbiosis, Legumes and Rhizobia, and finally, ChatGPT, an AI language model developed by OpenAI for providing assistance with creating content for this video. So goodbye, guys, and wish you the best of luck as you gain from this knowledge.